Good morning. It's 9 a.m. in San Diego, so we'll start the webinar. Thank you very much for your support of the webinar and for joining us today. Today's webinar is on the ICM 3D ligand editor, where we're going to learn how to optimize ligands, edit them, and also dock ligands. My name is Andrew Ori. If you have any questions after the webinar, please feel free to email or call. We're located in San Diego on the west coast of the States. And this is the first webinar in the series, and next week we have a webinar on structure-based virtual ligand screening. And then it's going to be followed by additional webinars on other forms of screening, docking and screening using, for example, induced fit, flexible docking and screening, covalent docking, and also fragment docking and linking and screening later on in June. If you have any questions during the webinar, please feel free to use the question bar on your GoToMeeting panel. And you could also raise your hand and I can open your mic at the end of the webinar if you have any questions that you want to ask. So with your registration, you get a free ICM Pro license for 30 days. You need to go to the support site, unpack the software, and then send me the 12-character host ID. And then a license key will be sent to you by email. It will run on Windows, Mac, or Linux. If you haven't received the license yet, please feel free to email again and send a reminder. But hopefully everybody has their license key already installed. The recording of this webinar will be available on YouTube along with a lot of other webinars that we have online there. If you just search for MolSoft on YouTube, you'll find the webinars. So today's topics include how to display and look at the interactions between a ligand and a receptor inside the ligand editor, how to edit the ligand in 2D and also in 3D. And then once you've made those edits, how to evaluate the effect of the modification. Is that a good modification or not? Then how to, you sit there and you can make as many modifications as you want. You can undo, redo. There's no concern about messing anything up because you can always go back to your original structure. And then you can save your edits to a fully interactive spreadsheet. So you can, if you make a ligand that you like, you can save it, and then you can just double click on that spreadsheet and it will load again when you reopen the project. Once you make edits, you may need to do a full ligand docking or minimization, and I'll show you how to do that. And there's also, in docking, you can include distance restraints and tethers to keep the ligand in certain place, maybe making a key interaction. And then in the ligand editor, you can also do covalent docking, fragment docking, and flexible receptor docking, but they will all be covered in the next few weeks as we go up. You can also, if you don't have a crystal structure, but you have some good chemical leads that are active, you can actually do design in 3D pharmacophore using MolSoft's atomic property field. So you just overlay the chemicals, and then you can design in those 3D fields. But there's a webinar online about that if you want to watch that, or I can send you that by email. Just contact me. So the ligand editor, it's been in development for over 12 years, and it was developed by MolSoft, but with great input from the medicinal chemists and the computational chemists at Novartis. So it's kind of designed to reward your chemical intuition, if you like. So it just enables you to load a structure or a complex, or you can actually load an apo structure and dock into it. But in most cases, you're in a scenario where you have a crystal structure and you have a good lead compound bound. And then the chemist can just start playing around with ideas to try and improve the ligand. You can delete atoms, you can change atoms very easily in 3D or in 2D, and then just commit those changes to 3D. And then you immediately see the effect of modification on predicted binding. And there's a paper from a couple of years now ago describing the interaction between MolSoft and Novartis and the development of this tool, along with other tools that they use at Novartis. That's in So a couple of years ago, using the docking tools and the editing tools in ICM, Max Totroff, our principal scientist here at MolSoft, was involved in the structure-based design of Webermir, which is it's a novel cyclic boronate beta-lactamase inhibitor, and it was a collaboration between MolSoft and the Rempex, which is part of the medicines company. If you're interested in reading more about that, the earlier design of the ligands was described in this paper here in Journal of Medicinal Chemistry. So today we're going to look at a crystal structure with a ligand bound. It's a kinase. It's in the family of cyclin dependent kinases. These CDKs, they alter the growth phase or state within the cell cycle, and this ligand we're looking at, called riscobatine, is in clinical trials for lung cancer and Cushing disease and other diseases. So we're going to be looking at that structure. We're going to start by understanding how to look at the key interactions between the ligand and the receptor. So this helps you in your design phase. So you would load the complex, and then you can display certain things like hydrogen bonds. You can display a surface shown here, for example, colored by a binding property, which could guide you into how to improve your ligand. You can display energy circles. These are shown here. This is quite a dramatic one here. This means that the red indicates that this group that you just added to your ligand is actually clashing with the receptor. So it's an easy way to observe whether your modification is any good or not. That's a clear example that it's not. The green circles shown here, that means that the energy of these atoms are decent, but here we have a problem. So you may want to avoid modifying in that area, for example. You can also display unsatisfied hydrogen bonds. That could give you an indication of where to build out to as well. And a key thing is also that you can display the relaxed ligand compared to the docked ligand. So when you, in any docking software, it's relatively easy to start building really beautiful molecules, but they're pretty not realistic. You may be adding artifacts, you're adding too much strain. So it's a good idea to not only keep it, we have a prediction of the strain of the molecule, which I'll show you, but you also can display it and compare it with the relaxed ligand. There's also tools for just visualizing the key interactions, which we'll show you in a minute. You can also work in the 2D environment as well, so not only can you see the interactions in 3D, but you can output a diagram like this, which shows the key interactions. So green shading represents hydrophobic region, blue is hydrogen bond acceptor, white dash arrows represent hydrogen bonds, grey parabolas represent the accessible surface area. So broken lines indicate there's a space here. And the size of the residue label, for example here, tryptophan, large, small, indicates the amount of contact between the ligand and the receptor. Okay, so before we get on to editing, we're going to look at the visualization tools. So if you have ICM open, you can try and follow along, or you can watch the video afterwards. But we will be working with, as I mentioned, that CDK kinase. It has a PDB code of 1 UNL. And if you watched the webinars from earlier this year, you can see that there's lots of search tools here. You can search Chembl, you can search PDB, but we're just going to search PDB by the code. So we're just going to type 1 UNL into this box. It's 1 UNL, and then you just press the search key, and that loads the structure. So we can see that now we have the structure in the ICM workspace. This region is called the ICM workspace. The terminal window, everything we do here can be scripted, but we will just close that for this webinar. We're not too interested in that. So we have 4 molecules, all colored. Only one of them has a ligand bound, that riscovatine molecule which I described earlier. And you can see it's riscovatine here. We can, if you want a closer look, we can just double click on riscovatine in the ICM workspace and then press center, and that will center the ligand here. So now we can orientate it to the way kinases are usually displayed, where you have the N terminal at the top here. We have the hinge region and the C terminal down here. If we just want a quick way of looking at the interactions between riscovatine and the receptor, we can just right-click on the ligand in the ICM workspace and then choose ligand pocket interactions. And that will show the key residues that are involved in binding, and we can also see that we have hydrogen bonds here with the hinge region of the kinase. So, but our aim in this webinar is to learn how to edit this ligand and try and improve these interactions. Maybe design a way into a space where we could maybe get a more specific inhibitor. And so we need to, for that kind of action, we can also redock. So for that we need to use the ligand editor. So you can see that, hopefully you can see at the top here that we have a number of tabs. The last one is called Lig Edit. So you click on Lig Edit, and then we have a number of buttons that are displayed here. So basically we first need to tell the ICM which is the ligand that we want to modify. So the ligand is riscovatine, it's here, and the first button is this one here. So you just click on that. In this case we only have one drug-like molecule in our PDB structure, and so ICM automatically finds it. It's here. It's, you can see it's 1UNL, and the PDB code for the ligand is ARRC, so it's found it. But in some cases you may have a ligand in each of the molecules of your PDB, so you need to choose which one you want to modify. Or you may have multiple crystal structures displayed. We can assign formal charges to this ligand by just clicking here. Or you can also, we're not going to cover it today, but you can also optimize peptides in the binding pocket as well. Just click here. And so we go OK. And now we can see we have another object in the ICM workspace. This is called 1UNL ligand. So it's basically copied the ligand from the PDB file, which is this object here, and that made another object. So now we can edit this ligand once we tell ICM the receptor. So the next button along is set up receptor. We click on that one, and it asks us which is the protein receptor. The protein receptor is 1UNL. In a couple of weeks' time we're going to work on flexible docking, so if you had multiple proteins with the pocket representing flexibility of the pocket, you can just select all of those structures and then you can dock automatically to all an ensemble of structures basically. But we'll cover that in a few weeks' time. Then we have some options. It's asking us, do we want to keep tight waters? Do you want to keep all waters? Or want to delete all waters? It's a kind of subjective question, so most, in a docking scenario, you would imagine that when you dock a ligand that you would displace the waters, but some are tightly kept, so you may have like 3 or 4 hydrogen bonds between the ligand and the receptor. So generally you would maybe choose to keep tight waters. That's a default. Then you need to optimize existing hydrogens and also optimize some other, so this is preparing the crystal structure for docking basically. In some scenarios you may have another software which you use that has prepared the protein already for docking. So it's a PDB structure, so it doesn't have any hydrogens, so we need to add hydrogens. We need to optimize the hydrogen bonding network to make sure that the orientations of the side chains are correct. There's also things that the crystallographer may not have seen, the asparaginins and glutamines. The orientation of those sides of those residues are kind of ambiguous in the X-ray density, so you need to optimize those for the best interactions. But we discussed that in previous webinars. So you can click all these. Mask missing side chains means that if there's a side chain that's completely missing in the crystal structure, then you can just not include it in the docking. If you uncheck this box, which is probably the better thing to do, but it's kind of subjective as well, ICM will build that chain into the structure. But in most cases, when you're optimizing a ligand, you're going to be using a high resolution ligand, so most of the side chains will be present. They would have been seen in the crystal, in the map. Okay, so there's also an option to find the binding pocket. If you look back in the webinars from earlier this year, we have a method for finding ligand binding pockets and allosteric pockets. You can do that, but we don't need to do that in this scenario because we already have the ligand in the binding pocket. So we just choose make box around existing ligand. And here you can see now we have this purple box around the ligand. So the default docking procedure, we build energy maps inside this grid, inside this purple box. And so you want to make sure the purple box covers the area that you want to build into. By default, it's usually a reasonable size, but if you've got maybe an elongated pocket and you want to build out from that ligand to that region, you may want to increase the size of this purple box, and you can just drag it like this in the corner. Okay, so once you're happy with that box, you just go OK. Now it builds the energy maps for docking, so it's building maps for hydrogen bonding interactions and other electrostatics and van der Waals interaction maps. This takes a little bit of time to run. It should be a couple of minutes, less than a minute usually. If it takes a little bit longer, I can load one I already prepared. Let's see. So we had a question: how do we handle metals in docking? So if you had a metal ion, if it's a common one which we have force field parameters for, zinc or magnesium, then you would just leave the ligand, the atom in the structure, and then it will be incorporated into the docking maps. Okay, it's taking a little longer than I expected. So I'm just going to load an example that I already have. Oh, it's finished. So when it's loaded, so this is what you'll see once the docking maps are prepared. So you'll see that in the ICM workspace, we now have another addition to our list of objects. We have op maps. So these are the maps where we're going to dock. Okay. So we're working on visualization tools initially, so we can investigate what the interactions are between the ligand and the receptor. So in the ligand editor tab, we can click and hold here, and these are most of the visualization tools. There's an option for pretty view, which will change the view a little bit. It's going to, you may want to play around with your preferred view, but this just shows the ligand this way. So in the ligand editor, we can you can display the receptor pocket, which I showed earlier. So you click and hold on this button here and choose receptor pocket. And it's colored by binding property. So blue, hydrogen bond donor; green, hydrophobic; white, lipophilic; red, hydrogen bond acceptor. It's colored that way. So you can see you can toggle the binding pocket on and off here, just clicking here. Another pocket you can display is called the ligand pocket surface. This is a little bit, it's kind of the opposite, it's colored the opposite of the receptor pocket, and it should give you a little bit better perception of where there is a cavity to bind into. So we can see at the back here there's no volume to build or to add any modifications to this ligand as in this pose in this region here. But if we rotate, we can see there's a gap here, and around this ring we have space here, and it's colored to guide you, and the coloring is colored to guide you basically. So it's saying that if you want to add a good group here, you need to add some kind of, it's colored blue, so you want to add some hydrogen bond acceptor in this region. This region's colored red, you may want to, hydrogen bond donor and hydrogen bond acceptor. So it's going to guide you in your design as well. So it's sometimes a good idea to leave this this surface on as you're making modifications. You can see that the hydrogen bonds are displayed here between the receptor and between the ligand and the receptor. You can toggle those on and off here. So the coloring of the hydrogen bonds are basically, the stronger they are, the thicker these spheres are, basically. And they're colored from green to blue, green being red being strong, and then blue being weaker. But you can play around with the parameters if you go to advanced and then choose preferences. You can play around and find weaker hydrogen bonds if you want to do that. Okay, so it may also, we can display unsatisfied hydrogen bonds, and you can see that you get these spheres where there's unsatisfied hydrogen bond region. You can display the relaxed ligand compared to the docked one. I won't show that. And you can also display the contacts. So if we click here, this is it's asking us about, so that when you display the contacts, it will make the residues that are making the bigger contacts, the residues will be thicker stick, and this is just a scale for that. So you just go OK. So now we can see that, for example, this residue here on the glycine-rich loop is making a very big contact. It's thicker. It's an isoleucine residue. We have now a table here which shows the contact area between this isoleucine is making the largest contact area, and it shows the distance and how buried that residue is. If you want to jump to another one, you can just double click here and it will select it, and you can just see, so if you center the center button, it will take you to your selection. So let's have a look at phenylalanine. It's yeah, it's at the top, it's at the back of the pocket, like the gatekeeper residue at the top of the hinge here, and it's making a big contact. Okay, so there's the other representations. This button here will show you those energy circles which I mentioned. It gives a value, so green is good, so we've got quite a healthy ligand at the moment. And if you want to look at the 2D interaction, you just click on the 2D diagram interaction button here, and that will generate, there's some cutoffs that you can use, but the default values are usually reasonable, but you can play around with that if you want to. And that will make that image, which we saw before, shown here. And you can right click on the image and save as image, or copy to clipboard. Okay, so these are the contacts. This is the 2D diagram. You may want to have display different levels of hydrogen. The hydrogens at the moment, it's set for, it's just displaying the polar hydrogen atoms. You can set it to say all atoms, all hydrogens, and all ligand and polar hydrogens of the receptor. So it's up to you how you want to play around with your hydrogen display. Okay, so we're going to move on to the editing side of stuff. So now you have your crystal structure. You may want to try and improve the interactions or make some modifications. So one way of doing that is in 3D, and you get this panel. I'll show you in a minute. So it's a panel, so you just click on here on the atom, and then click on the atom you want to change, and it will automatically change it. You can rotate torsion angles of the ligand using this button. You can choose one or more groups to modify. So you just click here, select multiple ones, and then select on the region you want to try, and it will automatically change those. It will sample those and give you a hit list of which ones you selected is better. You can erase, you can play around with bond types. These ones are for docking, and we can play around with some of these others later on in some of the other webinars, but those are the main things. So it doesn't matter what you do because it's full undo and redo capability. So we developed this for the chemists, and then we realized that actually chemists prefer to edit their ligand in 2D, which kind of makes sense. It's easier to do, I think. So if you want to edit in 2D, on this panel there's a button here. You set edit in 2D, click on that, and you will see this panel here, and it's like a regular ChemDraw sort of window that you can just change, modify the ligand in 2D. Okay. So when you make a modification, you can save it to a chemical spreadsheet, and the spreadsheets are pretty powerful. You can make predictions of molecular weight, polar surface area, Keiko binding, Herg binding, things like that, automatically on the fly. You can make plots, and so if you have a series of chemicals that you've made, you may want to look at the properties of those. You can export your ideas to PDB format, or you can save it as SDF file. You can export to Excel or HTML. You can also save it in ICM binary format, and then you'll be able to display them in the free ICM browser. So many companies that use ICM, they do that. They're computational chemists or they're med chemists. They use ICM Pro, which is the software you have, but then they share their ideas with their colleagues using ICM browser, which they have free on their laptops. Or you can display using IML View, which is our tablet software for molecular visualization. So you can share using different things. And in the chemical spreadsheets, there are lots of different features such as chemical searching, editing, clustering, plotting. There's some webinars on that also on our YouTube site if you want to look at those. Anyway, so let's go to that ligand, the rose-scovatine. So to start editing this ligand, I'm going to close this window. I'm just going to undisplay those. So as I mentioned, you can edit this ligand in 3D. So the edit tools are the next button along. This is set up ligand, set up receptor, and then the edit tools here. Okay, so you can, I'm just going to, just for the sake of, so you can delete atom, click on the eraser, click on the atom you want to delete. You see the hydroxyl there has been removed. And then you can evaluate the score, how good that modification is. So the lower the score, the better. So currently the score is -32. If we didn't like that modification we made, we can just return back to the original ligand just by erase this undo button. Now the ligand is back. So we can evaluate the score. You can edit bond types, atom types, just by clicking here and click on the atom and change it. Go back to the oxygen, just click here. We can change bond types as well, so we can maybe delete this. So use the erase button. And you just click on the atom type, and you can change it from single to double bond or triple bond just by clicking on the atom, on the bond. Sorry. So in here. So I'm just going to go back to the original ligand. Here. Okay. So we may want to make some modifications. So we can try, we can select one group here, for example, and just click on the atom that you want to try it. So you click here, and now it's the fluorine has been added to that region. You can undo it. So we can also do larger modifications. I'm just going to display that ligand surface, ligand pocket surface here. Okay, so we said that if you added something here, it's probably not a good idea because there's no space. So you can see that if we choose a larger group, just one larger group here, and we can add it. ICM will try and fit it. It's doing a small minimization. But I think we're going to see that the score is going to go very high. It's positive. And if we display those energy circles, it clearly flags that this modification, this region of that modification, is not too good. So it's making a clash with the receptor. So avoid that area. Okay, so if we look at this surface again, I'm just going to undo that modification. If we look at this surface again, we can see that around this ring we have some space that we can play around with to make modifications. So to do that, we can see we have some region here. Hopefully it transmits okay through the GoToMeeting, but we can basically we can select a number of different substituents. Just click on the panel here, and we can choose a sulfonamide, sulfonyl, and some halogens. So we select those, and then we go to this hydrogen atom coming off this ring. We want to try these substituents on this atom. So then we just click here, and it's going to, you can see in the bottom here it's saying try in groups. So it's doing a virtual screen of these groups to that position. And then you get a table of your hits. And it's ranked by score. So actually a bromine at that position is the most favorable, even better than the original hydrogen. So you can, if you want to just display that bromine, you can click here. So it's a hit list, so we can scroll down the hit list. So the next hit is the chlorine. We can see that pose here. And the sulfonyl group, you can see it here. The sulfonamide also gets a good score here. And the original fluorine, and then the original one actually comes, the hydrogen has a poorer score. But so if you wanted to load this structure in with the bromine, or let's just choose the sulfonamide group, just double click and that loads the structure in, and you can see the score. So if we like this modification, or we think it's reasonable, we can save it to a chemical spreadsheet. So there's a button here called Add Current Ligand to Chemical Spread to Table. So you just click on that one. So this saves that ligand in the spreadsheet. So it means that you can we can make modifications again to this ligand. Doesn't matter what you do, because you can always go back to this ligands table and just going to remove that surface and just double click, and that will load it back in. So you have it, always have it saved here. So if we want to look at, if we want to also keep one of the halogen modifications, we can just do the same thing. You can click and hold and tag it as well. So you can tag and add, say, series 1 or something like that, and that will give you another column in that ligand table with a tag. So you can also tag them. So then we can, in this table that we're storing our modifications in, we can just double click to load them. Or you can use this button here, and that just overlays the structure that you modified. Yeah, I've got quite a few questions I think I'm going to have to get back to at the end. I'll try to answer those at the end. So that's the 3D modification. To modify in 2D, as I mentioned, you just click on this 2D button here, and you get this window here, which you can just change. You can erase. You can use the templates here, or we have more complicated templates in these buttons here. Okay, there's also a chemical dictionary, so you just completely delete your molecule and just search for any molecule using the dictionary. So just aspirin, we'll load the aspirin there, or you can basically do it. So you can also just sketch a completely new ligand and then redock it. So you click this button here, which says apply changes to 3D, and now you can see that there's a little error there, but it's a minor thing. So the molecule is now, you now have the new the ligand bound. The modification that you made in 2D is projected to 3D. And so the key thing is to keep an eye on the score and the strain. So the lower the score, the better. The lower the strain, the better. Just for if you have a VLS score, you should use that. But some people don't have the VLS module of ICM and they just get this score. But they're both in the same scale, so you just need to make sure you concentrate on one or the other. So I've made a complete change to this. We may want to look at the history of what we've done in the modification. So if you click and hold on the undo and redo buttons, there's an option called modification history. And this will give you a list of everything you've done from, so we added a group, we loaded a group from the modifiers tab, we erased atoms, and this gives you a long list of all the steps we've just done. So we can always go back to the original ligand again by doing undo, 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 or you can use this and find it in the table in your modification history and then just double click and it will load that structure back. Okay. So as I mentioned, you can make modifications. If you make small modifications, then ICM does a small minimization to that, or you can run your own minimization by clicking here, and that will reduce any small clashes or any strained geometry. But you may want to redock the ligand, and I'll show that in a second. You can also modify the torsions yourself by if you want to try some different things, you can click on here, and then you go to the bond that you want to rotate, and you can see that the torsion angle is in orange. Hopefully it's shown, or the cursor changes when you hover over the, just one second, you hover over the bond. I'm not sure if that projects over the GoToMeeting. Anyway, you see the torsion angle, and you can just play around with rotation here or here. Wherever you click basically will rotate. So you see that you get a little handle on the bond which enables you to rotate that bond. So you can play around with torsion. If you want to redock yourself completely using ICM, we have the docking methods in ICM. So we're just going to talk about that briefly. So in the ligand editor, there's the full docking MultiSoft's ICM docking technology. So you can make a modification of the ligand and redock it. You can also load a SDF file and then dock that SDF file, so sort of small scale virtual screening. You can incorporate multiple receptor conformations, which we'll discuss in the webinar in a couple of weeks' time. But basically, you just need to load all the crystal structures that you want, and then you can modify your ligand. So you could load different subtypes of your receptor and try and design subtype-specific ligands, for example, or you may have a crystal structure of a drug pocket which is kind of flexible, where a loop comes in and out. Then you can use those multiple receptors or multiple conformations to design and screen. You can refine the side chains. You can allow certain residues to be flexible during docking, which we'll cover in a few weeks' time. And as I mentioned, you can also, if you don't have crystal structures but you have good leads, you can superimpose those lead compounds and then design new ones in the 3D pharmacophoric fields in the same way as we're doing. There's covalent docking, fragment docking, and using tethers and restraints, which I'll show. So ICM has been performing extremely well in the D3R Grand Challenges, which are a industry-wide docking competition. Basically, it's a completely blind competition. The crystal structures and the ligands are provided by the pharmaceutical industry, and we don't see those until the results. This was led by Max Totroff here at Molsoft, our principal scientist, and Polo Lam and Ruben Upagayan. There's a couple of papers describing the methods, the docking methods, but ICM has been ranking in first place for the ability to redock a ligand and also prediction of binding energy, but you can read about those methods in this paper here. So to redock in ICM, to redock this ligand, we can click on the next button along. If we want to minimize it, so first we need to look at the score. So yeah, I was playing around with the torsions, so we've got all these clashes and it's showing us. Earlier I was playing around with the torsion, so I want to go back to the original ligand. Hopefully this will be the one that's slightly, it's probably this one here. Okay, so if you want to minimize this ligand, we just click on minimize button. That will do a very small minimization. But if you want to do a full docking, you would go here and redocking the ligand. So it's currently docking the molecule. It's using the ICM bias probability Monte Carlo method to randomly sample the conformation of the ligand in the pocket. It's assessing, it's jumping around the pocket, finding good energies and putting those into a stack, and it will output the results in a second. So once you've docked, you can change this, but by default the number of conformations is 5. So you can use this square button here to just toggle those conformations in the pocket. Just going to change some of the original. So they're ranked by score. So the lower the VLS score, the better. So the top one is -39. And if we go down here, we can see some poorer scored ones. You see they're all docked in the pocket. So we can uncheck those, and that's the ligand docked. If you had an SDF file, for example, in the distribution, there's an SDF file, and you can just dock that immediately to that pocket by going to dock table, and that would dock, and you'll get a hit list similar to this of multiple ligands, but this is just the one ligand. We've just redocked it. If you're doing virtual screening, then everyone who has the license, you have all these tools. So you have homology modeling, you have all the cheminformatics tools, and they also have the docking menu, which enables you to do virtual screening in a more efficient way. So the ligand editor is more for one-on-one design, but you can do virtual screening if you wanted to. So if we're redocking, we've made in a scenario, maybe we've made a modification over here. We know that these rings need to be in place because they're making these key interactions with the hinge region here. They're making hydrogen bonds. So we can tether or keep these in place when redocking. If we want to tether them, we click on this T button here, then click on the atoms that we want to tether. Maybe we want to keep this nitrogen in place. So just click on T, and so now we have 3. You can see the red crosses through the atoms. You can also see we have a new table with docking restraints. So it's telling us that this atom is restrained, and we can toggle those on and off. We can use them or not use them. It's up to you. You can also set a distance restraint. So we can say we want to always keep this hydrogen bond, so we need to keep a distance restraint between this backbone oxygen and this hydrogen. So if we click on the D for distance restraint, you can click here on one atom, click here on another, and you can now see in the table we have another option called distance. And so we can choose maximum and minimum distance for that. So we can say maximum distance would be 2.2 and minimum distance 1.8, for example. And so now if we redock, this ring system won't move. And probably anyway, if you redocked it, it would find that conformation pretty well. Anyway, so you may not need, for kinases you probably wouldn't need to do that, but for maybe other targets you may need to add some kind of distance restraint if you want to do that. Okay. So that's the docking. You can also use a template docking as well, so you can have a key template, and you can use that. This kind of a more advanced use of the software. So you'd read in a template substructure, and then you would dock to that region. Okay, so those are the key tools. Some of the other things that you can do is you can add new substituents to this table. So you would just click Add New, and you can sketch something that you want to. There's something you regularly use. You can just add it, and it will be included in the. No, it's just a made-up thing. You can add it to that. So you just close it, and then say yes, and then that's added to the panel here, and then you can just use that for docking regularly. You may want to screen these substituents plus a lot of other fragments to a particular point. In ICM we have an inbuilt database, so you could say find group, and you can screen. It's basically a collection of all these groups plus a few other ones, or you can use your own set of chemicals called use external table, read in and dock those. So basically all you do is just click on hydrogen and click run, and it will screen all those substituents in that one place, and you will see a results table like before. So you would have a hit list basically here of ranked by score, which substituents are preferred, and you can just double click on those and it will load it into the ICM workspace, into the 3D space. Or you can toggle display on and off, so you get a hit list. So other options in this advanced menu, we'll show you this in a couple of weeks' time, but you can select certain residues to be in the pocket to be fully flexible. You can refine the whole pocket. You can also do a bio-isosteer scan, which I can send information, but basically we built, Eugene Rausch, who's main cheminformatician here, has built a bio-isosteer database from Kemble and also from the PDB, and it will find, if you select a group, it will find the best bio-isosteer that makes a good interaction. You can also do, these are more on the fly things that are done in the background, but you can rather than interactively, but you can also do multiple position scan. So you can just select the groups that or select all of them, and it will try at each point, each hydrogen, which and dock them at each point and see which position is better for your. So it'll basically give you a hit list similar to this, but it will go through the whole ligand trying to find. It's basically an R group scan. You can also try do hetero scan as well with different atoms, and that will just run in the background, and then you'll get a hit list similar to this for the preferred R groups. Okay. So I just want to thank you very much for joining the webinar, and thank you for your questions. I've got quite a few to go through. I'll try and go through them, but if not, I will get back to you by email. The next webinar is next week, which is on virtual ligand screening. So that will be how to prepare your protein structure for virtual screening, where to get large databases from to screen, how to perform the screening, and then how to analyze the hit list results at the end, and how to cherry pick chemicals for experimental testing. So that will be on May 16th. So this enables you to screen larger databases to a pocket